This is lecture 19 on chapter 19, titled Eukaryotic Genomes, Organization, Regulation, and Evolution. As an overview, we note how eukaryotic genomes work and evolve. In eukaryotes, the DNA protein complex, called chromatin, is ordered into higher structural levels than the DNA protein complex in the prokaryotes. In this illustration, actually it's a photomicrograph, a light microscopic photomicrograph. There's no scale bar, but this is one strand of DNA along the length of a chromosome. It's the DNA in a eukaryotic chromosome from a developing salamander egg. So you can see in this fluorescence micrograph, you get a sense of the complex organization of the chromatin in a eukaryotic chromosome. Part of the chromatin is packed into the main axis, where it looks kind of white, of the chromosome, while those parts that are being actively transcribed are spread out into the red loops. So this kind of fits the illustration that we had been doing in prior lectures. We draw a squiggly line where it's condensed, and then an open straight line where it was being expressed, and then back to a squiggly line. Well, this is how it actually would look on one unduplicated chromosome of a homologous pair in a somatic cell. Okay, so this is not undergoing mitosis or meiosis. This is an actively expressed chromosome. Whatever genes on it are being expressed in a somatic cell. Oh, wait, I'm wrong. We said developing salamander egg. Okay, well, so it's got to be in the S phase where we have gene expression before the DNA replication would begin then, right? So an unduplicated chromosome in a somatic cell living its life would look the same way. This is what a chromosome looks like when the DNA is unwound enough to allow for gene expression to occur, but it's differential gene expression. Not every gene on this chromosome would be expressed. But we have to remember that in the cell cycle, the S phase is when we'd get the gene expression for protein synthesis to sustain the rest of the cell cycle, when the chromosomes are replicated and condensed as the duplicated chromosomes. Okay, so packaging of the chromosomes is part of what we're going to discuss in the beginning of this chapter. So both prokaryotes and eukaryotes must alter their patterns of gene expression in response to changes in environmental conditions. It's a key part of homeostatic mechanisms, keeping things the way that they should be, like the thermostat on the wall keeps your house at a constant temperature. But also, obviously, as we progress toward chapter 1 to 21, there are changes in gene expression through the development of an organism, too. Also, remember, it's internal and external environmental conditions. If your body gets hot on the inside because of a fever, certain changes will occur. If your body gets hot on the inside due to the outside, like we're getting here in Texas, where it's April now, then when you're just sitting in the heat, or if you're in hot water like a sauna, then, well, it's not the sauna that's hot water, but you know what I mean. External environmental high temperatures generating internal environmental high temperatures, then there might be a completely different pattern of gene expression responses, differential gene expression responses. And then obviously during embryonic development there's a pattern of gene expression that is coordinated with the overall development of the organism. Okay, moving on. Concept 19.1. Chromatin structure is based on successive levels of DNA packaging. Packing. Good enough. Eukaryotic DNA is precisely combined with a large amount of protein. So it is a highly organized sequential series of packing or packaging of DNA with protein to produce the condensed chromosomes, as you would see in the case of the duplicated chromosomes. That's really the only time, unless you can think of something different, the only time that we would have the complete condensation of the DNA within a chromosome. Eukaryotic chromosomes, on the other hand, I challenge you to think of when it would be the least compacted. Are you done thinking yet? I would predict that it would be during DNA replication, right? Have to. Wouldn't have to be all unpackaged and disassociated with protein all at once, but it would undergo the most complete unpackaging or unpacking throughout its entire length during the replication events. Okay, eukaryotic chromosomes contain an enormous amount of DNA relative to their condensed length. And of course, the textbook gives all these numbers for lengths and distances and all that stuff, and it's kind of neat, but I'm not going to walk around remembering that stuff in my brain. I got plenty of other important things to remember. How about you? So nucleosomes, or beads on a string, are important basic units of the packing of the DNA. So the proteins associated with the DNA at this level are called histones. They are responsible for this first level of DNA packing in chromatin. They bind tightly to the DNA, and the association of the DNA and histones seems to remain intact throughout the cell cycle. So regardless of the degree of compaction or looseness, or packed state and unpacked state, these proteins are staying right there. They may come off for a moment, and then they're right back on as soon as they can. In electron micrographs, unfolded chromatin has the appearance of beads on a string. We see that in this lower panel on the right-hand side. I don't see a scale bar for this. It may be that this scale bar, no, this is a much higher magnification than that. So this scale bar would have to be shrunk down to not much bigger than the tip of my pointer right here next to one of these nucleosomes, as they're called, or beads along the chain. So when the DNA is stretched out basically as the linear double helix, there's also some shortening due to this, right? If you it's like a spring. If you pulled on each end, you could stretch it out farther so it was straight linear. But here, as the helix, there's really the initial shortening of the DNA. So with the electron micrograph, it would look a bit like this. We see the 2 nanometers scale here, and the lower panel on the left is showing the nucleosomes, basically the 10 nanometer height, if you want to, or width of the fiber, the DNA fiber. So what we're seeing here are the histones collected together in here, and then at the tips of these arrows, the histone tails. There are linker DNA or strings in the necklace, if you want to think of it that way. This says bad here, and that should say bead, like the beads on a necklace. The nucleosome is the bead, and then the linkage DNA in between the nucleosomes is the strand. So this linkage DNA is what's being shown up here. So 1/5 the diameter, we could say, even if you want to go to that. So there's a five-fold or 5 times increase in the thickness, you can use that term, of the chromosome due to the formation of the nucleosomes. So this is the first level where there's the protein interaction causing the packing. So the DNA and histone molecules form beads on a string, the extended chromatin fiber seen during interphase. A nucleosome has 8 histone molecules with the amino end tail. We see those sticking up. That's the amino terminus of the protein, or the amino end tail of each projecting outward. A different type of histone, H1, we see it here, can bind to DNA next to a nucleosome where it helps to further compact the 10 nanometer fiber. Higher levels of DNA packing occur as we proceed. The next level of packing forms the 30 nanometer chromatin fiber, so we are now 15-fold greater in thickness, 3 times the histone effect. So here what we are seeing is the 30 nanometer fiber as an illustration and an electron micrograph, kind of wandering around a little bit, but we can see it here on the right. The string of nucleosomes, you can see the 4 little histones and the I think it's 2.5 wraps of the DNA around them. The nucleosomes coil. The string of nucleosomes, so the string, coils to form a chromatin fiber that is 30 nanometers in diameter. The tails are not shown on the histones, but they're sticking out. And this form is also seen during interphase. Next, the 30 nanometer fiber in turn forms loops or domains made up of a 300 nanometer fiber. So I'm not going to keep doing the fold increase, but you can see that we're getting thicker by leaps and bounds as we get shorter. The linear length, overall length of the DNA double helix strand is going to decrease as we compact it. So here we see the protein scaffolding on the bottom in this illustration, and the looped domains of the 300 nanometer fibers. So it's obviously the average diameter of this structure. So here we see the protein scaffolding at much higher magnification. We're zooming in on just an area like this, and you can just barely make out the loops. You can definitely make it out in the actual photograph in the textbook. The loops are sticking out like this. In a mitotic chromosome, a duplicated chromosome, so the way they seem to be approaching this illustration is that they started with a chromatid on a duplicated chromosome in interphase. That's when the condensation is beginning. Do you remember exactly which stage of interphase? I'll leave that for you to try to recall. Well, I can't remember either, so if you do, you beat me to it. I'd have to go look it up. Okay, so the loop domains themselves coil and fold, forming the characteristic metaphase chromosome. So now we see these 300 nanometer loops forming these crinkles in them, if you want to think of it that way, more than doubling. And then here we see the transmission electron micrograph. And then ultimately we zoom back in magnification. We were just looking at this little chunk here, and as we predicted, it is a sister chromatid on a duplicated chromosome that we were following through interphase to get to the metaphase chromosome. Obviously, this would have appeared like this in prophase as well. And pre-metaphase, it would be like this. But anyway, this is the characteristic metaphase chromosome that we would see in a karyotype, correct? And it is its overall width, we can't really say diameter now, overall width would be 1,400 nanometers. And here we see the actual transmission electron micrograph of one. So there we are with our duplicated chromosome, the sister chromatids here, one of a homologous pair. So our challenge now in this chapter is to follow along with the authors and correct or insert duplicated or unduplicated chromosome and say, are we talking about a mitotic duplicated chromosome or a meiotic duplicated chromosome, or are we talking about an unduplicated chromosome throughout either of those types of cell division, say in the haploid gamete, or are we talking about unduplicated chromosome within a somatic cell living its life and doing its job? So if you pay close attention, you'll see the authors don't always state exactly. They leave a bunch of words off and hope that within the context of what follows the word or precedes or follows the word chromosome, we can insert the appropriate descriptors so we know exactly what we're dealing with. So we're starting to build on that intensity where everything we've learned in the past is now coming in for us to put it in place and to know exactly what someone is talking about when they just use, for example, the word chromosome. Okay, in interphase cells, most chromatin is in the highly extended form called euchromatin. So U means true, so the true chromatin all stretched out, the most highly unpacked or extended form that it could be. And we already said that that's because it has to undergo replication. So it has to lose all of its protein, as replication is proceeding down the length. Remember the replication forks, and there can be multiple bubbles along the length of the human gene. So wherever that bubble forms and you've got the replication forks going away from each other, all these proteins we're talking about here, especially the histone proteins, have to let go for a minute and then come back immediately when the new strands are available and bundle it all up again. Concept 19.2, gene expression can be regulated at any stage, but the key step is transcription. All organisms must regulate which genes are expressed at any given time. During development of a multicellular organism, so developmentally differentially regulated gene expression, excuse me, during development of a multicellular organism, its cells undergo a process of specialization in form and function called cell differentiation. So as we proceed, keep in mind for multicellular organisms these two basic types of differential gene regulation, beyond developmental. There is tissue-specific gene expression. Remember, tissues are collections of different cell types, like an epithelial cell type, like the epithelial cells on our skin. And then connective tissue, well, the epidermis is an example of epithelium. Let me use another tissue, okay? That one wasn't working. Let's think of the liver. What is happening to me here? Go to connective tissue. You've got different cell types making up the tissue. You can have connective tissue cells and immune cells there. My brain's not working for a good example, I'm sorry. Okay, its cells undergo a process of specialization in form and function called cell differentiation. So if your mind's working a little bit clearer in mind right now to think of a good example of tissue, then let's do brain tissue. That's a good one. You've got neurons and glial cells, so at least two different types. So you have tissue-specific gene expression for it to be nerve tissue, and then you have cell-specific expression for getting a neuron versus an astrocyte, a type of glial cell. Okay? Another example would be pituitary tissue. You can have tissue-specific expression for that to be glandular tissue, and then you can have cell-specific expression so that only one cell type is going to make growth hormone, and another cell type is going to make the hormones that control the gonads, the gonadotropins. So hopefully that helps you to establish these two terms, tissue-specific gene expression and cell-specific gene expression. As a particular cell is undergoing differentiation, it first has to establish for the type of tissue it's going to be its tissue-specific gene expression, and then if it is going to become part of a cell population with a very particular task in that tissue, it will undergo cell-specific differentiation. It's very important conceptual conglomeration there for understanding differential gene expression in multicellular organism. So each cell of a multicellular eukaryote expresses only a fraction of its genes, those that are specific for the tissue that it is within, and those that are specific for its particular cellular function, tissue-specific and cell-specific gene expression. In each type of differentiated cell, a unique subset of genes is expressed. So they're trying to get at these two terms, tissue-specific gene expression here and cell-specific gene expression here. And even in my graduate course, I always trick the masters and even most of the doctoral students with a question where I say, is this true or false? The growth hormone gene is found in every cell in our body. And many of them say false because all they can think about is when they were studying growth hormone gene expression in a particular cell subset of cells within the anterior pituitary. And yes, every cell in our body contains every single gene that we have in our genome, but it only expresses those that are essential for it to do its job. Okay, I'll get off my soapbox there and let's move on. Many key stages of gene expression can be regulated in eukaryotic cells. So let's follow through this typical illustration of gene expression and note that regulation can occur all along the length. So now when we are progressing to this global utilization of the term gene expression, we are thinking of the title of our previous chapter, was it? No, the one before that, from gene to protein. So the full expression from gene to gene product, the protein. And we're following the expression of a gene from its transcription to the synthesis of a protein and the protein living its life cycle and being destroyed, the final step here, for a for a gene that is transcribed for ultimate production of protein. Okay, so remember, other genes can be expressed only for the RNA, and then the RNA does the job, has its particular function for the biology of the cell. Okay, so let's follow through. Here's a signal coming to a cell. So now we're at a somatic cell, right? Unless this is a signal for the reserve cell to undergo mitosis or to trigger meiosis. Anyway, a signal comes to the cell and be interphase cell. So here's the chromatin. The chromatin modification would be the DNA unpacking involving histone acetylation and DNA demethylation. So those are important modifications to the DNA and the proteins that pack it to free up the DNA so that it is available to be expressed. The gene will then become available for transcription as the double helix, and that gene will undergo transcription to produce the primary transcript, giving us the RNA, and here we see the different colorations. The reds that you can see are representing the exons, and the ones that really don't show up, kind of a, I don't know, pale orange or something like that, are the introns. Then there is the RNA processing, 5' cap, poly A tail. Where'd my pointer go? Poly A tail, and the exons have been spliced together. So this is the mature mRNA in the nucleus that will translocate to the cytoplasm. The term they use is transport. Don't know if it's physically transported, guided through some cellular action, or just diffuses. Probably has cellular guidance to do it. mRNA is then in the cytoplasm after it's come through a nuclear pore. It's going to live its life and also fight the challenge of being degraded. We talked about mechanisms for delaying degradation and even storing the protein within the cytoplasm until needed. This happens in the female gamete, the egg. But translation can occur throughout the lifespan of the messenger RNA, hopefully before it's degraded. Then the polypeptide is produced. Then the cleavage, chemical modification, transport to the cellular destination, so post-translational processing. You then have, you can have an inactive protein toward the end of that. Once it is active, it's in the active pool of protein, and it lives its life, does its job, and dies. So there will be degradation of the protein, and then you have the degraded protein. If these are individual amino acids that haven't been damaged along the way, they can be recycled as raw material for this. Same with the nucleotides here. If they're still in good shape, they can be recycled, right? So at any point along the length, you can have modification of gene expression. Thinking of starting with a signal for the gene to be expressed to the point where the protein, the gene product, has lived its life and is degraded. Then, regulation of chromatin structure can be related to this. Genes within highly packed heterochromatin are usually not expressed within cells. So back when we studied the cell, we talked about euchromatin and heterochromatin within the nucleus. We said the euchromatin is more unwound and is being actively expressed or is available to be expressed to one degree or another. And the heterochromatin was where it was packed tightly together. The gene expression molecules can't get to the DNA. Those genes are turned off. Histone modification is part of what must occur for this process of making the DNA available or unavailable for expression within a particular cell. Chemical modification of histone tails can affect the configuration of chromatin, and thus gene expression. So here in this little illustration, we are reminded of chromatin changes leading to transcription, RNA processing, mRNA with its degradation, translation as well, part of its life cycle, and then the protein processing, function, and degradation. So we're at this level of regulation, chromatin changes. Here we are reminded of this illustration of the, was that 10 nanometer fiber level, a nucleosome here, and we take a high mag look at the nucleosome, and here's what we're seeing. We are seeing a blue, green, yellow, and red histone with its amino terminal tails sticking out radially toward the periphery of this, right? And these amino terminal, or these tails, the amino terminal is just the very tip of it, right? So these tails have amino acids available for chemical modification. So the histone tails protrude outward from a nucleosome. In this end view of a nucleosome, each type of histone is shown in a different color. The amino acids in the N-terminal tails are accessible for chemical modification. Histone acetylation is one example. In acetyl group, a COCH3 chemical formula can be added to the tails. So the the left here we see the unacetylated histones, and when they are acetylated, it seems to loosen the chromatin structure and thereby enhance transcription. Now in the textbook they talk about different charges of the proteins and various characteristics that can be altered to allow this to happen. We don't need to focus on that level, but it is still quite interesting because we know enough to understand it. Let's just try to learn enough so that we can remember some of it, perform well on the exam, and feel good about the volume of information that we will place into our working knowledge. Am I trying to cheer you on here? Must be. So acetylation of histone tails promotes loose chromatin structure that permits transcription. A region of chromatin in which nucleosomes are unacetylated forms a compact structure on the left in which the DNA is not transcribed, the heterochromatin. When nucleosomes are highly acetylated, the right, the chromatin becomes less compact, euchromatin, and the DNA is accessible for transcription. Another modification, DNA methylation. Addition of methyl groups to certain bases in DNA is associated with reduced transcription in some species. So I would assume that this could occur or be in state within both euchromatin and heterochromatin. Epigenetic inheritance is a characteristic of the transmission of traits, and is the inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence. So it may involve some of the things that we've discussed where the chromatin has been modified. Regulation of transcription initiation is the next idea we want to talk about. Chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. So we've had some introductions to this so far. We've talked about promoter regions and transcription factors and so on and so forth. So let's get into the detail a little bit. Now here let's note that we are considering control at the level of transcription and RNA processing. So associated with most eukaryotic genes are multiple control elements. Segments of non-coding DNA that help regulate transcription by binding certain proteins. So here we see start site would be right about here where the dark blue begins. Downstream to the right, exons, introns, and then the poly A signal sequence, and then somewhere a little bit further downstream, the termination region for transcription, and the translation termination would be in here somewhere too. I guess it would have to be up here in this last exon before the untranslated 3' untranslated region, right? I mean, there's a 5' untranslated region in the first exon and a 3' untranslated region in the last exon. Okay, that's not labeled here. Let's just move on. The promoter is here, upstream, immediately upstream, and then farther, as we might have up to 1,000 or so nucleotides in the promoter. Then you start getting 1,500, 2,000, 2,500, 4,000, something like that. Upstream, you can have proximal control elements. They're close, or they approximate the promoter. And then those that are distant or distal control elements. And these are typically an enhancer, and they're way far upstream, as I had noted. The primary transcript is shown here. We remember that. It's the pre-mRNA, 5' to 3'. Did I have my 3', 5' correct up here? Keep an eye on me. I get that reversed. So after transcription, the initial transcript, we've got the exon, intron, exon, and then the poly A signal. And then the cleaved 3' end of the primary transcript will occur. Then there will be RNA processing, the 5' cap and the poly A tail added, introns excised, and exons spliced together to give the coding segment between the start codon and the stop codon. And then the 3' untranslated region, I mentioned that, and the poly A tail that also does not get translated. Upstream of the start codon, you can use the upstream thing wherever you give your reference point, as long as you say, if I start at my home and go upstream, right? So upstream of the start site is the upstream of the start site for transcription is the promoter. So upstream of the 5' of the start codon, that's for translation, right, is the 5' untranslated region. So it's on the messenger RNA before the start codon to initiate the first amino acid to be placed into the growing peptide, and then the 5' cap. So all of these steps are potential targets of regulatory mechanisms for controlling this global gene expression that we're considering, from changing expression rate at the gene to the death of the protein made by ultimately from that code within the gene. So the roles of transcription factors to initiate transcription is the next idea we want to well, just the roles of transcription factors, next idea we want to think about or consider. And first under that is their role to initiate transcription. So eukaryotic RNA polymerase requires the assistance of proteins called transcription factors. We already know that. Enhancers and specific transcription factors will act at the proximal control elements located close to the promoter, or at the distal control elements groups, which are called enhancers. So they may be far away upstream from a gene, or even in an intron within the gene, right? Within the coding portion of the gene. So let's consider first the example of an activator shown here. It's a protein that binds to an enhancer and stimulates transcription of a gene. So let's follow through this. Hopefully you're looking at the figure in your text or in the printed slide. Get our orientation. Here's the TATA box within the promoter, and then the downstream gene portion of that. And then way upstream is the distal control element, an enhancer, 3 little gold bars there, and the activators, proteins that can bind to that. So activator proteins, number 1 here, bind to distal control elements grouped as an enhancer in the DNA. This enhancer has 3 binding sites. And then step 2, a DNA bending protein. Imagine that was all you had to ever do in your life, huh? A DNA bending protein brings the bound activators close to the promoter. Whoop. Other transcription factors, mediator proteins, and RNA polymerase are hanging around in the corner of the drugstore just waiting to get their transcription initiated, right? So they're all there. And then step 3, this line going way over. So here, as we cross it, we know we're obviously talking about regulation at the level of the gene transcription. 3 tells us that the activators bind to certain transcription factors and mediator proteins, helping them form an active transcription initiation complex on the promoter. We had studied the components of the transcription initiation complex before. And when that complex is completed, the RNA polymerase gets up and goes for RNA synthesis. Some specific transcription factors function as repressors, so we want to be able to turn on genes and turn off genes, as well as, so to regulate, but as well as to modulate how much on or how close to off they might be. And timing, when should they be on and when should they be off. Those types of events are occurring all the time in our cells. It's a good thing we don't have to sit around and think about it, right? So some specific transcription factors, even though we are right now, but we can live without thinking about it, right? Probably through the semester you've been saying that a lot. I could live without having to think about this. Some specific transcription factors function as repressors to inhibit the expression of a particular gene. So there might be points where we need to turn off a gene completely, although we might want to turn it back on again later when we would need it. So some activators and repressors act indirectly by influencing chromatin structures. So here you see these terms, indirectly, directly. Always pay attention. You know I love those kinds of questions. Coordinately controlled genes is a next idea that we want to consider. So unlike the genes of a prokaryotic operon, coordinately controlled eukaryotic genes each have a promoter and control elements. The same regulatory sequences are common to all the genes of a group, enabling recognition by the same specific transcription factors. So we also have series of enzymes involved in biosynthetic pathways, anabolic, catabolic. Those genes are not in tandem on a chromosome. They may not even all be on the same chromosome. But what they do all have is common regulatory sequences, so that when the transcription factors or whatever other proteins are involved in affecting the expression of one of those genes in a cell, they all get expressed. So the cell-specific gene expression, right? And as well, the ones that regulate and modulate on-off, and how high does the rate of expression go, and how low does the rate of expression go, those are all going to be shared as well. So you get a coordination of the expression of these genes that are involved in these complex interprotein biological processes. So mechanisms of post-transcriptional regulation are now considered. An increasing number of examples are being found of regulatory mechanisms that operate at various stages after transcription. RNA processing, well, we talked about alternative RNA splicing already. The different mRNA molecules are produced from the same primary transcript. They got it right here, didn't they? Not on the exam question that you'll see from the earlier chapter, but we corrected that in our presentation. Depending on which RNA segments are treated as exons and which as introns. So we considered this before, and we just see an example here. So now we're considering regulation at the level of, regulation at the level of RNA processing. So we see the DNA with the exons and introns, and then giving them some different colors there. And then the primary transcript, and I guess this is the guy we're following here because it doesn't end up in the final mRNA. Actually spliced it into two populations of mRNA rather than one we considered, where you just leave out an exon or two and put it all together, and then they just disappear. Anyway, another variation on the theme. End up with what looks like, I guess, two chunks of mRNA. So mRNA degradation, in the next level we want to consider. The lifespan of mRNA molecules in the cytoplasm is an important factor in determining protein synthesis in a cell. So how long do they sit idling, protected in the cytoplasm before they are actively transcribed? How many ribosomes jump on them and transcribe the heck out of them while they're available? And how long are they available to do that before somebody comes along and chews them up, catabolizes them, and they're no longer available? So, the lifespan is determined in part by sequences in the leader and trailer regions of the molecule. RNA interference by single-stranded microRNAs can occur, and this is at the level of mRNA degradation. And, of course, if you impact degradation, accelerate it or slow it down, you impact translation. So we see both of those boxes lit up here in our little cell that we've been following. So the microRNAs can lead to degradation of an mRNA or block its translation. So let's follow this illustration along. I think they bring in the numbered things as we go. Oh, there's one. The microRNA, miRNA, precursor folds back on itself, held together by hydrogen bonds. There's our friends, the hydrogen bonds. An enzyme called dicer moves along the double-stranded RNA. It's double-stranded here where it just folded on itself, cutting it into shorter segments. With 3 here, we see the little segments coming along, and then a protein complex coming in to become associated with the miRNA. Somewhere along the way, the hydrogen bonds get broken, so it's single-stranded. So one strand of each short double-stranded RNA is degraded. The other strand, miRNA, then associates with a complex of proteins. The bound miRNA can base pair with any target mRNA that contains the complementary sequence. And finally, the miRNA protein complex prevents gene expression either by degrading the target mRNA or blocking its translation. The ribosome can't get past it. So initiation of translation is the next level of regulation we want to consider. The initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to specific sequences or structures of the mRNA. Alternatively, translation of all the mRNAs in a cell may be regulated simultaneously. And we're just trying to highlight some of these ideas. They're explained a little more detail in the text. You're welcome to read that to get the additional detail. So protein processing and degradation is the next level of regulation. After translation, various types of protein processing, including cleavage and the addition of chemical groups like a glycoprotein, are subject to control. Now we jump to that last stage, the degradation. So proteasomes are giant protein complexes that bind protein molecules and degrade them. Here they don't bring in the numbers, they just bring in the whole thing. Somebody got mixed up, I guess, but anyway, they did it for us. I didn't have to do it. That's what's important. So now we are considering regulation at the level of protein processing and degradation, more specifically degradation. So first we see this protein to be degraded. It's got alpha helices in it, and maybe just got 3 helices in it, right? And then this other protein, the ubiquitin, that will bind to it, and then you have a ubiquitinated protein. So multiple ubiquitin molecules, I like saying that word, ubiquitinated molecules are attached to a protein by enzymes in the cytosol. And then a proteasome comes along. The ubiquitin-tagged protein is recognized by a proteasome which unfolds the protein and sequesters it within the central cavity. So something happens to a protein within its lifetime to where its character has changed such that it's probably not going to be functional any longer or at all anymore. and it has an affinity for ubiquitin. Once it's ubiquitinated, then it will be gobbled up by a proteasome. Whoops. I was waiting for my little number to come in there, but it's already there. The proteasome and ubiquitin will be recycled, and the protein fragments, the peptides, will be present for further degradation and recycling for the amino acids that are still good. Some might be damaged and will have to be destroyed completely. Enzymatic components of the proteasome cut the protein into small peptides, which can be further degraded by other enzymes in the cytosol. So this is a process that can be regulated. How rapidly do you remove proteins from the active pool? Concept 19.3, cancer results from genetic changes that affect cell cycle control. The gene regulation systems that go wrong during cancer turn out to be the very same systems that play important roles in embryonic development. Types of genes associated with cancer are the genes that normally regulate cell growth cycle and division during the cell cycle, and it includes genes for growth factors, their receptors, and the intracellular molecules of signaling pathways. Research on tumor viruses led to the discovery of oncogenes and then ultimately proto-oncogenes. Onco comes from the Greek tumor, so these are genes associated with tumors. They are cancer-causing genes. They have that capability. Proto-oncogenes are normal cellular genes that code for proteins that stimulate normal cell growth and division. So a DNA change that makes a proto-oncogene excessively active converts it to an oncogene which may promote excessive cell division and cancer. So it's important for you to understand that when a cell or a group of cells becomes cancerous, it's not only that it changes its regulation of the cell cycle genes, so its division is out of control, right? The cell division is out of control. As we talked about in an earlier chapter, mitosis. But expression of a lot of other genes can change too. So for instance, there's an oat cell lung carcinoma, from which you begin to get expression of genes for many of the hormones that are typically produced by the anterior pituitary gland. So the symptoms that a patient with an oat cell lung carcinoma presents with first might be associated with an overactive adrenal gland. And they might suspect first there's a problem with the anterior pituitary, but an astute physician would think check for oat cell carcinomas because very commonly they get the gene for the ACTH, adrenocorticotropic hormone, that drives the adrenal cortex, and they just pump it into the blood. They just make tons of that hormone and pump it into the blood. So right now we're considering the cell division, but be aware that other genes within the newly formed or forming cancer cells can be dysregulated as well. So here we're seeing genetic changes that can occur, that can turn proto-oncogenes into oncogenes. So first, translocation or transposition. The gene moved to a new locus under new control, and you get normal growth stimulating protein, but in excess. Here we see gene amplification with multiple copies of the gene. They're now basically separated a bit, but they're in a series, not quite tandem, I guess. And you get still normal growth stimulating protein, but in excess. And then the next possibility is you get point mutation within a control element, and you get the oncogene itself with normal growth protein normal growth stimulating protein in excess also. So these three, the endpoints are the same, but the in-between points are different, based upon different set of events. And then the one all the way over on the right here, we have a point mutation within the gene, changing it to an oncogene, such that you have a hyperactive or degradation-resistant protein. So it persists longer and sustains its chemical signal within the process of the cell that it affects. Next, we can consider tumor suppressor genes that encode proteins that inhibit the abnormal cell division. So you can either drive your car, you can either slow your car down with letting your foot off the gas pedal, or you can put the brake on, right? And you can convert proto-oncogenes to oncogenes and accelerate your car's growth rate, if you want to think of it that way, by pressing on the gas pedal, or you can take your foot off the brake. So if something happens to a tumor suppressor gene, taking your foot off the brake, then it can allow disinhibition of the cell growth. If it normally inhibits abnormal cell division and it becomes compromised, then you have disinhibited abnormal cell division. Interference with normal cell signaling pathways is the next topic to consider. Many proto-oncogenes and tumor suppressor genes encode components of growth stimulating and growth inhibiting pathways, respectively. So the RAS protein encoded by the RAS gene is a G protein that relays a signal from a growth factor receptor on the plasma membrane to a cascade of protein kinases. And this is the way it works normally for cellular responses to growth hormone. Let's follow through with this. What we're considering here in the brown box is the condition where a mutation might have occurred, producing a hyperactive RAS protein, product of an oncogene, that issues signals all on its own, independent of the normal mechanism of the ligand-gated membrane receptor. Remember, we talked about these before. Growth factors binding to two monomers, then they form homodimers, and then the phosphorylation occurs, part of which leads to a cascading effect of the activation of the RAS protein, activating protein kinases, ultimately affecting transcription factors in the nucleus, ultimately affecting gene expression, ultimately affecting a protein that stimulates the cell cycle. So when it's regulated, you have regulated cellular division. Excuse me. When it is dysregulated, you have dysregulated cell division. I'll let you read this legend in the textbook or in your printed copy of the slide to follow through this, but we already know enough to understand it, right? We've studied this stuff before. Oh, there's number one. Growth factor, receptor, G protein, protein kinase phosphorylation cascade, transcription factor, and we're on to the next one. Another example, the cell cycle inhibiting pathway. So the previous one was the cell cycle stimulating pathway. We should have noted that. This one is an example of cell cycle inhibiting pathway. So the P53 gene encodes a tumor suppressor protein that is a specific transcription factor that promotes the synthesis of a cell cycle inhibiting, actually, of many, plural, of cell cycle inhibiting proteins. So let me click and see if they're bringing in numbers here. Okay. So number one, the DNA damage in the genome occurs. So DNA damage is an intracellular signal that is passed via. Number two, the protein kinases and leads to activation of the active form of P53. The activated p53 promotes transcription of the gene for a protein that inhibits the cell cycle. The resulting suppression of the cell division ensures that the damaged DNA is not replicated. Mutations causing deficiencies in any pathway component can contribute to the development of cancer. So here in the brown box, mutation, defective or missing transcription factor such as p53 cannot activate the transcription of the protein that inhibits the cell cycle. So if a cell goes bad, one bad apple can spoil the batch unless you get that bad apple out of the basket to begin with. So mutations that knock out the p53 gene can lead to excessive cell growth and cancer. We see that illustrated here. The effects of mutations, increased cell division, possibly leading to cancer, can result if the cell cycle is overstimulated, as in A, or not inhibited when it normally would be, as in B. So the effects of mutations can be proteins overexpressed or the cell cycle overstimulated, leading to increased cell division. The phenotype otherwise can change too, as I told you before. So there's new chemicals that might be produced by these cells. They are just focusing on the cell division that's out of control. And if a protein is absent and the cell cycle is not inhibited, bad cells can spoil the basket, right? The multi-step model of cancer development is the next idea we will consider. Normal cells are converted to cancer cells. We've considered a few ways that can occur. Remember, there's environmental chemicals and things like that that can change the genes of cells and make them cancerous too, but everything taken into account, normal cells are converted to cancer cells by the accumulation of multiple mutations affecting proto-oncogenes and tumor suppressor genes. A multi-step model for the development of colorectal cancer is an example of this. So we start on the left and go to the right. So there's the colon. Here's the normal colon wall, illustration of a section through the wall. The normal colon epithelial cells are seen here. And less of the tumor suppressor gene APC, or maybe something else, can bring about a small benign growth or a polyp. And then activation of the RAS oncogene, or loss of tumor suppressor gene DCC, can lead to larger benign tumor and adenoma. Just the fact that this is called an adenoma tells me that it's because adeno means glandular, so it's probably going to be pumping out some types of chemical messengers. You get a loss of tumor suppressor gene p53, additional mutations maybe, and end up with a malignant tumor or carcinoma. And then you see it beginning to infiltrate nearby tissue and spread. It can get into lymphatics and all kinds of remote tumors can be instated. Now, certain viruses promote cancer by integration of viral DNA into the cell's genome. So it can be troublesome just like that. And we've studied viruses. We understand how this integration of the viral DNA into the cell's genome occurs. Next, inherited predisposition to cancer is a topic to consider. Individuals who inherit a mutant oncogene or tumor suppressor allele have an increased risk of developing certain types of cancer. Now on to concept 19.4. Eukaryotic genomes can have many non-coding DNA sequences in addition to genes. The bulk of most eukaryotic genomes consists of non-coding DNA sequences often described in the past as junk DNA. However, a lot of evidence is accumulating that the non-coding DNA plays very important roles in the cell. So we don't just disregard it anymore, we at least mention it, and maybe someday you'll become a scientist and study some and figure out what some of it does. Or maybe you won't, right? The relationship between genomic composition and organismal complexity is the next idea we want to consider. So compared with prokaryotic genomes, excuse me, the genomes of eukaryotes generally are larger, they have longer genes, and contain a much greater amount of non-coding DNA both associated with the genes and between the genes. So now that the complete sequence of the human genome is available, we know what makes up most of the 98.5% that does not code for proteins, ribosomal RNAs, or tRNAs. We see that in the pie chart here. Start with the skinniest one first. Exons, the region of genes encoding for protein, tRNA, rRNA is about 1.5%. Plus the 98.5, so the rest of the pie is making that up. The repetitive DNA that includes transposable elements and related sequences, about 44%. ALU elements, don't know what it is, 10%. Simple sequence DNA, 3%. The repetitive DNA unrelated to transposable elements is about 15%. With a large a part of that being the large segment duplications, 5 to 6%. Unique uncoding DNA, about 15%. And then introns and regulatory sequences, about 24%. So you don't need to know any of those names and numbers, of course, but it's just interesting to see the distribution of these various types of DNA, making up 98.5% of the DNA that we pack away and do all those things with. While we mentioned the transposable component, let's consider transposable elements. We talked about it with prokaryotes and related sequences for eukaryotes. So the first evidence, we mentioned this at the end of Chapter 8. You see now that between edition 6 and 7, they shifted it forward into the next chapter. Whatever. So the first evidence for wandering DNA segments came from geneticist Barbara McClinton's breeding experiments with Indian corn. So here's a picture of her. She's all happy at her microscope, noting her observations. A couple of ears of corn here with the different colors in it. So we've all seen this type of corn, the Indian corn. Hang it up for Halloween or whatever we do with it, I guess. The movement of transposons and retrotransposons is a concept to be considered. Excuse me, a topic within this concept. Eukaryotic transposable elements are of two types: transposons which move within a genome by means of a DNA intermediate, retrotransposons which move by means of an RNA intermediate. So let's take a look at this illustration to see what sense we can make of it. In the upper portion here we see excuse me, transposon movement, copy and paste mechanism. So the DNA of a genome is shown here. And this is the transposon in the darker blue color. We note this diagonal break here, so they're talking about a remote distance. The transposon is copied. It becomes a mobile transposon, and then somehow is inserted as a new copy of the transposon somewhere else in the genome. Down below here, excuse me, we're considering a retrotransposon movement, and we see the DNA of genome, the retrotransposon, RNA made from that, a reverse transcriptase that acts upon it and makes it double-stranded or amplifies, making the double stranding. In panel B, the movement of retrotransposons begins the formation of a single strand of RNA intermediate. The remaining steps are essentially identical to part of the retrovirus reproductive cycle. In movement of transposons, so we see RNA to make DNA, then make double-stranded DNA, then it gets inserted as a new copy of the retrotransposon. Retro is referring to making mRNA, and then retro back to DNA, get double-stranded DNA, and then that can be inserted somewhere else within the genome. So in movement of transposons by the copy and paste mechanism, and in movements of retrotransposons, the DNA sequence remains in the original site as well as appearing in a new site. Sequences related to transposable elements. There are multiple copies of transposable elements and sequences related to them. They are scattered throughout the eukaryotic genome. In humans and other primates, a large portion of transposable element-related DNA consists of a family of similar sequences called ALU elements. Oh, here it is. Still not going to tell us about it, and that's fine with me. Other repetitive DNA, including simple sequence DNA. Simple sequence DNA contains many copies of tandemly repeated short sequences. So, in eukaryotes, there are instances of tandem association of genes, and these are tandemly repeated short sequences. So it's common in the centromeres and telomeres, where it probably plays structural roles in the chromosome and really doesn't have anything to do with protein synthesis, protein expression. Genes and multi-gene families, this is what I thought I was describing in the prior slide, but got a little screwed up there. But you should be able to clarify that. Those are just structural. Here's the genes in the multi-gene families. So the multi-protein complex biological processes within a cell, like a biosynthetic pathway with many enzymes. We've seen these before. Most eukaryotic genes are present in one copy per haploid set of chromosomes. The rest of the genome occurs in multi-gene families, collections of identical or very similar genes. Some multi-gene families consist of identical DNA sequences, usually clustered tandemly. Okay, so that does occur tandemly for certain instances, as I'd started to describe prior slide. So genes that do get expressed all the way to the level of protein can occur in tandem in eukaryotes. It's not as common as in prokaryotes, such as those that code for the RNA products. So this illustration, is showing part of the ribosomal RNA gene family. 3 of the hundreds of copies of ribosomal transcription units in a salamander genome are shown at the top. TEM, transmission electron micrograph. Each feather, you see how it looks like a feather, corresponds to a single transcription unit being transcribed by about 100 molecules of RNA polymerase, the black dots, the dark dots along the DNA, moving left to right. So shorter where the polymerase has been on there a shorter period of time, and longer where it's been on there for a longer period of time. The growing RNA transcripts extend out from the DNA. In the diagram below, this electron micrograph, one transcription unit is shown here. It includes the genes for three types of ribosomal RNA, 18S, 5.8S, and 28S. Adjacent regions that are transcribed but later removed in yellow. Here and here and here and here. A single transcript is made and then processed to yield one molecule of each of the 3 ribosomal RNAs, boom, 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 in red, which make up part of a ribosome. Here we find them in the large, containing the 28S and the 5.8S ribosomal RNA subtypes, and the 18S within the small subunit. A 4th ribosomal RNA, a 5S RNA, is also found in the ribosome, but the gene encoding it is not part of this transcription unit. The classic examples of multigene families of non-identical genes are 2 related families of genes that encode globins. So hemoglobin comes back to visit us. We see the beta globin subunit. There's 2 of those. There's the heme group. And the alpha globin subunit, there's 2 of those. So it's a heterotetrameric protein in its quaternary structure. Right? Hetero for different, tetra for meric for parts or units. So 2 alpha and 2 beta globin subunits. So on the left here we see the beta globin gene family on chromosome 16. There's the oh, I don't recognize my Greek alphabet. Shame on me. Okay. You'll let me off this time, won't you? There's a bunch of different looking things here, so different genes, right? And again, what we're looking at are the genes on a template strand of a non-duplicated chromosome, one of a pair, one chromosome of the homologous pair. I got it out there eventually, right? And then there's fetal and adult types that are shown here. And then the beta globin gene family on chromosome 11 in the embryo. Well, there's epsilon, I recognize that. And the fetus, and then the adult. So we get developmentally regulated differential gene expression of the beta globin gene family, as well as the alpha globin gene family. So the human alpha globin and beta globin gene families, hemoglobin is composed of the 2 alpha, 2 beta globulin polypeptide subunits. The genes, the dark blue encoding alpha and beta globins, are found in 2 families organized as shown here. The non-coding DNA separating the functional genes within each family cluster includes pseudogenes, shown in green. You see them here and there, and then right here. Is that psi, that funny-looking Y thing? I don't know what it is. Non-functional versions of the functional genes. Genes and pseudogenes are named by Greek letters that I obviously don't know too well, huh? Concept 19.5, duplications, rearrangements, and mutations of DNA contribute to gene evolution. The basis of change at the genome level is mutation, which underlies most of genome evolution. The topic then, duplication of chromosome sets. So there can be accidents in cell divisions that can lead to extra copies of all or part of a genome, which may then diverge if one set accumulates sequence changes. Duplication and divergence of DNA segments are related to unequal crossing over during prophase 1 of meiosis. And this can result in one chromosome with a deletion and another with a duplication of a particular gene. Let's follow this illustration. We recognize our homologous pair the non-sister chromosomes, chromatids, excuse me, and the high mag here, transposable element shown there, a gene, and I guess that would be another transposable element. And we see a similar type of a thing below on the non-sister chromatid. If there's an incorrect pairing of two homologs, they got stuck together in the wrong spot during meiosis, then you end up with a long chunk here and a short chunk there. So you have a short limb and a long limb chromosome within the homologous pair. Excuse me. So this illustration is showing gene duplication due to unequal crossing over. One mechanism by which a gene or other DNA segment can be duplicated is recombination during meiosis between copies of a transposable element flanking the gene. Excuse me. Such recombination between misaligned non-sister chromatids of homologous chromosomes produces one chromatid with two copies of the gene and one chromatid with no copy. Two copies and no copy. Excuse me. Evolution of genes with related functions. The human globin genes come back again, not necessarily hemoglobin, but there's a lot of different globin genes, globin proteins even, like immunoglobins or immunoglobins. The genes encoding the various globin proteins evolved from one common ancestral globin gene, which duplicated and diverged. So we see it starts here with the ancestral globin gene, and over evolutionary time, long time, right? Duplication of the ancestral gene, then mutation in both copies, then transposition to different chromosomes, and then further duplications and mutations, so that ultimately, this one is looking at the hemoglobin, but there are other globin proteins. Ultimately leading to what we observed in the previous slide, the earlier slide. Now, sequence duplications of these genes and random mutations gave rise to the present globin genes, all of which code for oxygen-binding proteins, for the alpha and beta globin protein families. The similarity in amino acid sequences of the various globin proteins supports this model of gene duplication and mutation. So the alpha globins, or globins, and the beta globins, or globins, I'm not going to bother with the numbers on this table. Is that something you want to do right now? I don't know about you, but if you've been with me throughout this whole hour and 20 minutes, had enough of that stuff. So let's wind it up with just a brief comment on the evolution of genes with novel functions. The copies of some duplicated genes have diverged so much during evolutionary time that the functions of their encoded proteins are now substantially different. We saw the alpha-beta globins still contribute to oxygen transport. Rearrangements of parts of genes, the exon duplication and exon shuffling, a particular exon within a gene could be duplicated on one chromosome and deleted from the homologous chromosome. So in exon shuffling, errors in meiotic recombination lead to the occasional mixing and matching of different exons, either within a gene or between two non-allelic genes. So this figure is showing the evolution of a new gene by exon shuffling. Exon shuffling could have moved exons from ancestral forms to the genes for epidermal growth factor, fibronectin, and plasminogen, shown on the left here. Here's the epidermal growth factor gene with multiple EGF exons, and the fibronectin gene with multiple finger exons, shown in orange, and the plasminogen gene with a Kringle exon in blue. What fun these geneticists have naming things. Portions of the ancestral genes here may have contributed to the formation of the TPA, or tissue plasminogen activator gene as it exists today. It's got a little bit of the fibronectin with the exon shuffling, a little bit of the epidermal growth factor, and a little bit of that Kringle. Where's Chris on this? Kringle but no Chris? Well, Merry Christmas. The TPA gene may have been formed. So the order in which these events might have occurred is unknown. Duplication of the Kringle exon from plasminogen after its movement could account for the 2 copies of this exon in the TPA gene. Each type of exon encodes a particular domain of the TPA, tissue plasminogen activator protein. How transposable elements contribute to genome evolution. Movement of transposable elements or recombination between copies of the same element occasionally generates new sequence combinations that are beneficial to the organism. Some mechanisms can alter the functions of genes or their patterns of expression and regulation. And we will end there. I appreciate your time and attention in listening to